Hi again. So what Lauren asked me to talk about this time around is what you're looking for in your microbial community. And I have a lot of thoughts on that maybe are a bit different from what you've heard in other places, but it's good to get a diversity of ideas to support a diversity on your farm. So the other thing that I have spent the last 5 years looking at are ways to make testing more accessible on the ground. And I actually really like some of the soil health testing methods that NRCS promotes because they don't take expensive tools. They don't take a lot of time and coordination to take samples, send them off to somebody, and then wait for those results. And as far as waiting for results, I chuckle sometimes at the number of growers who will approach me because they know I know a little bit, enough to be dangerous about interpreting soil tests. And they'll bring me soil tests that they've had in their pickup truck for 2 or 3 years because the extension agent told them they should take a soil test. And they've never, they wonder if I can interpret it for them. So they've never called back the lab. They've never had anybody go over the test with them. And it doesn't help your soil much if you don't do that. So if you're going to take the test, make sure you can interpret it. And so one thing I like about learning to do it yourself is that there's a lot that you can figure out on the farm with your own observations, and you don't have this gap between what was tested and what you interpret. So let's look at some simple, affordable ways to monitor specifically microbial communities on the farm. And of course, a lot of these methods will cost less, and the more you use them, the more you can get the cost down. So if you're doing microscopy, you might have to buy a microscope, and that's going to take a little chunk of change. But a good microscope will last your lifetime and probably get your kids started on the farm as well. You might decide to upgrade it because there's neat new technology out there, but it's not necessarily going to improve your results. Doing it yourself is usually a lot faster, which means you can make your decisions faster. And you can customize it to the questions you have. Imagine filling out some of these vegetable farmers that are market gardeners that have 100 different crop species out there. Well, your crop's not going to be worth the cost of taking 100 different plant sap analyses, and yet you know that what you measure in your tomatoes is not going to inform your asparagus very well. So you can adapt your own if you're doing it on the farm. And you can sample as often as you'd like. Time and practicality will dictate some of this. But I think when people start new management practices, the microbial analysis can be real helpful. One benefit to microbial analysis is that the microbes respond very quickly. So it might take a while to see a change in your soil aggregation. It might take a while to see how quickly water percolates into the soil, but within literally hours or days, you can see changes in your microbial community. Wonder how tillage is really impacting your soil? Take a profile the day before, go in a day after, and you'll see a difference. Does the cover crop really change your soil health? Well, you can see changes in microbial diversity as that cover crop is germinating. And does that great new bug in a jug that the vendor is selling you really work? This is probably the single most common question I get from growers. Is this stuff any good? Well, if you can test your own, you can try it on your soil with your crop and have answers very quickly. So actually, 6 to 24 hours after I add an amendment, I can see changes in the microbial community. And so these are some of the advantages to learning it. Now, what's affordable for do-it-yourself testing really depends, again, on what you're growing and what you're trying to achieve. And so having options lets you choose the strategy that works best for your place and time. If your budget looks kind of like, you know, the shoestring, the easiest thing to look at is indirect monitoring. Don't even look at the microbes. Look at the visible critters in your soil. Look for the insects, look for the beetles, look for the earthworms, okay? Some ways you can, some of the ones that these guys eat microbes. You're not gonna find a tiger walking through the Chihuahuan desert because there's not enough food to support a tiger. And you're not gonna find an earthworm, which is a fungivore, in a soil that doesn't have any fungi in it. So if you're not seeing earthworms, you probably don't have soil fungi. Pill bugs, okay, springtails, all of these. Now, some springtails can be very small. Some of these are better with the microscope. This pleasing fungus beetle is one that's a little more common in forestry or orchard type lands. But these are just a few examples of visible critters you can look at that will tell you what your soil microbial community is doing. So you can literally take the shovel that we've heard many times today is a good indicator of soil health. You can dig up a shovelful and look to see what's living in there. If you want something quantitative, you can use pitfall traps. Just take your empty can of beans from home, a little tin can or a solo cup or something, dig a hole, put the cup at the level of the ground, and leave it out in the field. Go back the next day and see what insects fell into it. Okay, then you can actually count. You can determine how many of these you want to put across the field. You can monitor, you know, get 5 or 10 pitfall traps, count, and you've got lots of real statistically reliable count data that you can use to go as deep as you want to go quantitatively. And you're still talking about that. You're still falling within that shoestring budget, okay? A slate test is one of the NRCS tests we see promoted often. And this is kind of funny. I could not for the life of me find a good picture of this. I really didn't have time to go take a good picture of this. I tried to see if AI could interpret a graduated cylinder and a clump of soil. And you get the idea, but it's a little odd looking, okay? For a very small budget, I actually offer a class on an online program called Udemy that shows you everything you need to know about how to buy a microscope, look at the soil under the microscope, and distinguish some of the different kinds of microbes. And that's actually, I'll be honest, it's in need of an update because I made it in 2018. But I keep saying I'm going to get on there and update the course, and maybe this summer will be the magic summer. But there are some pros and cons to using a microscope. How many of you in here are already using a microscope to regularly monitor your soil biology? 1, 2, okay, a couple of people. Like I said earlier, I started out down this journey thinking every farmer needs a microscope. Of course, I'm coming in from this scientific microbiology background. And you know how spouses just have to be so practical and call you to reality when you've got great ideas? My husband David was constantly telling me, we need to start our own farm so that you can experience some of this and know what you're asking people to do. I don't use the microscope as much as I thought I would. You don't have time. So the advantage of using the microscope to check your soil health is it gives you a very direct measure of microbial abundance. Today, A lot of people recognize that we need that soil biology in there. Even 5 years ago, to me, the biggest power of having a microscope, I would bring it with me to conferences and show people what was in there. The biggest advantage was that's when you realize your soil's alive. When you see these little things wiggling past the objective in the microscope, you know, oh, my soil's alive. And before that, it's just dirt. If you want to measure the microbial diversity, you can directly measure, at least at the kingdom and phylum level. Now, some people who want to spend hours learning to recognize microbial morphologies can get a little bit more precise than that, but at least at these higher taxonomic levels, you can get a rough estimate of your microbial diversity. You can do all this in-house. A microscope doesn't take a huge footprint. You can keep it in the closet most of the year and take it out from time to time when you've got questions about your soil biology. A same-day analysis is possible. And once you've paid, I mean, you could probably get your microscope and a few supplies you need for under $500. Certainly for $1,000, you'll be set for life. And then the cost of each sample is pretty nil. Okay, less than $1 a sample. The downside is, like everything else worth doing, it takes learning what you're doing, and it takes a little bit of a time commitment. You can't get to the fine levels of diversity that you can with a test like PFLA or a DNA test or something of this sort. It's a little bit labor intensive. I have found that to get, a friend of mine, David Johnson, does a lot of his own microscopy and has assured me that with 12 samples, 12 replicate samples, you can get like within 99% accuracy of your microbial count and diversity measurements. He can make it very reproducible. I can't even make myself look at 12 samples on the same day under a microscope. It starts hurting my eyes. I get distracted. I don't have his tenacity. I'm not doing it. Okay, so I don't get 99% accuracy. I do 5 samples. But the other thing you'll see is that your results vary user by user. And so you need to establish your method and then stick with it. Otherwise, your numbers are going to be all over the board. So the goals when you're looking at your microbial population are to look for diversity and abundance. We've heard all day long, biodiversity, biodiversity, biodiversity. Well, you want that in your microbial community as well. Now, when we go back to that first shoestring budget, a real quick shoestring budget to estimate your microbial diversity underground is to look at the plant and animal diversity above ground. Okay, if you're growing a monoculture, 600 acres of soybean, you are not going to have the diversity that a multi-species cover crop will have underground. So you can estimate some of that just by looking at what you're growing. I teach microbiology now to pre-nursing students, and the thing I challenge them with all the time is this idea, as we've come to understand the human microbiome better, that disease is not caused by too many germs. We see their textbooks are telling them, wash your hands, put sanitizer, sanitize everything, here's the best chemicals to sanitize with. And I like to say disease is actually not caused by having too many germs there, it's caused by having too many of the same kind of germs there. And the only way you get that purity is if you've really disrupted the ecosystem. That's when you get imbalance. So if you've got that above ground diversity, you'll have that below ground microbial diversity. One reason, when you're fertilizing, one reason I like biological fertilizers better than chemical fertilizers is because, again, if you're eating all the same kind of food all day, if you only eat bread and water diet, you're not going to have much diversity in your gut microbiome. Okay? Same thing for your plants. If you're giving them this pure chemical to eat, they're not going to have the diversity of food that they need to trigger production of different phytochemicals that attract or repel different microbes. So the abundance, of course, is part of the story. You want lots of microbes there, but you don't want a lot of the same kind of microbe. That's a sign of disease. So you want to keep that population both diverse and abundant. When you have that rich microbial population in the soil, you're going to see the soil doing a better job of extracting nutrients from parent material. There are actually, we have found fungi embedded deep in caliche, 80% caliche soils. You can dig pit holes in there, pull samples from down deep, look at them under the microscope, and you will see little hyphae from microfungi that are pulling nutrients out of that caliche. Fungi transport a lot of water. And just like a lichen, the fungi in a lichen serve to support the algae in a lichen with water. I believe that in our dry climates, these underground fungi are working to transport water to the plant roots. So they can help move that water and nutrient into the plant. They can convert atmospheric nitrogen and carbon into food. Just like we have plants carrying out photosynthesis, we have many cyanobacteria and microalgae that can convert that nitrogen and carbon into plant food. I'm sorry, the plants are converting the carbon, not the nitrogen, in case I tripped on that a little bit. The microbes can build more soil. That's the essence of that O layer that Jay was talking about. And they minimize pathogens. Now you say, wait a minute, how can having all these microbes minimize pathogens? Because one eats the other. So when you have diversity, you're going to have, you're never going to build up too many of the same kind of something. You're always going to have one thing that eats the other. So I was asked to show some different kinds of microbes and what they look like. And if you're looking at prokaryotes, which are the largest microbial population, remember that all plants and animals make up, and fungi, and eukaryotic microbes like protists, make up a single taxonomic domain. But we have two domains of prokaryotes. We have bacteria and archaea. And they are basically the smallest specks you will see under the microscope. So all you're really going to be able to distinguish at that level is the shape. If they're spherical, we call them coccus. If they're rod-shaped, we call them bacillus. If they're rigid spirals, we call them spirillum. If they're long, flexible, stretched out spirals, we call them spirochetes. And if they're comma-shaped, we call them vibrio. There's some names you can give them that aren't that important if you're just doing this for yourself, but based on how they group up, what is important is that you realize if you see something like these brown staphyloclusters and the diplopairs, they might both look coccus, they're both round shaped, but they're probably a different species if they're aggregating differently together. And so you can count those as diversity. These filamentous microbes, actually, if you see something green and filamentous like this, celebrate. You've got photosynthetic cyanobacteria in there. And those are capable not only of fixing carbon, but also of fixing nitrogen, at least if you haven't over-nitrified your soil. Let's see, the sarcina are kind of cube-like clusters, and strepto, think of strep throat, streptococcus bacteria form these long chains. Okay, so the shape of the aggregate and the shape of the bacteria are how you tell them apart. Now fungi are going to be much larger, so you can see much more detail in them under the microscope. And I've put a handful of the more common shapes that you might see here, okay? So penicillium has these kind of broom-shaped structures under the microscope. If you are lucky enough to get a whole fungus that's forming these fruiting bodies, okay? The fusarium, which we think of as a bad guy, but again in small numbers, it's going to be transporting water and nutrients to your plants, okay? So it's going to have these kind of club-like shaped structures that you might see, and they tend to break off. Sometimes you'll see individual spores. In fact, the penicillium individual spores actually just look like little balls, bigger than the bacteria, bigger than a coccus bacteria, but they look like little balls. The alternaria has this very clear divided shape in the spores. There's an aspergillus down here that also looks kind of broom-shaped or brush-shaped. And the germinating hyphae in the middle tells you this thing is reproducing, it's alive, it's sprouting, okay? The budding yeast here is really drawn out of proportion. It's going to be much smaller than the other ones that I've shown you here because yeast tend to be single-celled, but you may see these large structures with an internal nucleus. Let's see if yeah, there we go, okay. So this would be the cell nucleus, and if you can see something, first of all, if it's large enough to even see whether or not it has a nucleus, it's probably a eukaryotic microbe, okay? Bacteria are too small to see that kind of distinction, at least if you're looking at under a light microscope. Now, if you happen to have an electron microscope, that's a different game, but you're not going to buy that for $300. Okay, protists are another class. These are basically one-celled eukaryotic organisms. They don't form the long hyphae or strands that you might see with the fungus. But again, if you see green, you've got something photosynthetic. And algae are no longer classified as plants. We call them protists. And you'll see the cell nucleus in all of these, okay? And the green algae, if it's got lots of little legs on it as it moves around, that's a ciliate. And if it's got this odd shape that changes as it goes, these always remind me of Far Side cartoons because Gary Larson was a big fan of the microbial world. And that would be something like an amoeba, okay? So the mobility, of course, comes from having the cilia around it. The cilia themselves are hard to see under the microscope, but you'll see this little waving motion, and it really will dart across your objective. And if you're looking at things at 400x, the bacteria are little specks that are hard to even see, but these other things are going to take up a big part of your slide. They might take 20 or 30% of your slide area of your viewing field through the objective. You'll see a lot of plant residues. These might be kind of greenish or they might be brown, but you can distinguish them as plants because plants have very rigid cell walls that tend to look not always perfectly square, but clearly very divided, okay? And so you'll see decayed plant material in there. And I often get asked, what is this stuff? Well, it's just kind of a rotting leaf or root or something. Okay. You're also going to see animals. So really, you should see every taxonomic kingdom in there. Okay. You'll see nematodes that look like little worms, and you can learn to distinguish them by mouth shapes, learn if they're fungivores or plant-eating nematodes or whatever. Tardigrades are fun, and I've only seen a few of these in samples. These are amazing little creatures that look kind of like koala bears, and they are vegetarian, but these things are tough. They can take heat, they can take cold, they can take, imagine Sheldon Cooper. These guys will do advanced physics experiments with tardigrades, putting them into quantum entanglement that I don't even understand. But they've taken them up into the space station. Tardigrades are just tough, okay? Springtails, of course, are real popular in soil food web discussions, and they eat a lot of the other microbes. And this is why I say, if you have a lot of everything, there's always going to be something that eats something else and keeps the numbers in control. And that's why when you have ample diversity, it's really hard to create disease. So if we put this all together, I actually had some videos here, and unfortunately, oh, they've got them working. So let's see if can I turn them on from here though? I don't know, lots of times clickers won't do that. This is actually what I saw under my microscope. So you've got some decayed material here that's just a cluster of decaying plant material, but this long stretch here, there we go, is a fungal hyphae. And you can see you have to move the slide. These dark divisions are real common in what we call ascomycete fungi. These are your common molds like aspergillus or penicillium that are sometimes called dark septate fungi because they have those divisions. But this thing is crossing several slide views. So these hyphae are what is binding those soil aggregates together and creating networks that communicate with your plant and transport nutrients from underground. Now the little specks here, that's what you're going to see of bacteria. So don't sweat trying to tell, is this E. coli or is this Pseudomonas or what? Just worry about, are they different from each other? Are the shapes different? Are the sizes different? Are the clusters different? Because that's about as good as you're going to go. Okay, let's see if this will switch. I actually lost my camera on my microscope and learned that you can get quite a bit from your cell phone camera looking through the objective, okay? So this gives you an idea of the dynamics you'll see and, you know, as these things move around the stage. And I had some cool music on there, but I don't think that's coming out. The next one is a bigger, it's actually a tardigrade. And I was so excited when I saw this little guy because he did a little dance for us. There we go. So your soil is alive, okay? And remember, he is flattened by a slide when he's doing his thing there, okay? So you don't get quite their full shape because you're squashing them with a cover slip, but you do get some idea of what's going on. Now, the last thing that I've been playing with to measure soil diversity in real time is soil respiration, and there are a lot of single soil respiration methods that are used widely now that you can use. I'm using something called substrate-induced respiration with several substrates. And this is a little bit more complex. It's something that would take some training and time to work out. But what it measures is the functional diversity. Instead of looking at what microbes are in your soil, it looks at what the microbes are doing in your soil. Can they break down proteins? Can they break down fats? Can they break down fiber? Can they break down lignins and cellulose? And we've got it down to where we can get answers within 24 hours. And so one thing I like to use this for is the whole bug in the jug question. Is this stuff any good for the soil? Okay, it demonstrates the microbial ability to cycle nutrients. It rapidly detects changes in microbial populations. It can be used in place or in the presence of these soil amendments. So you get your soil sample, you divide it equally, you add the amendment to one batch, leave the other alone, you put them through the test, and overnight you can figure out whether your soil microbial diversity is increasing or decreasing with that amendment. Now, phase 2 of this research that I'm still looking forward to is to make sure that every time we see a change in microbial diversity in the soil, you're seeing some kind of measurable improvement over time in the plant. That's kind of a big ask in terms of research with the one-person lab, but we'll plug away with it and hopefully attract some partners and see where we can go with that. Okay, and I am getting the red light, so I already know what it's like to get pulled off the stage here. I'm good at overstaying my welcome. But thanks a lot.